the HRM week 1 to week 6 full review. Start off with the blood composition. There are going to be three main functions of blood: transport, regulation, and protection. What are we going to be transporting? We're going to be transporting the nutrients, gases, waste products, hormones, and enzymes. And obviously we have to regulate body fluids, acid-base balance, water balance, and body temperature. And we have to protect the body, so immunity, hemostasis, blood proteins. And what are the components of blood? So you're going to have two. You're going to have plasma and formed elements. Formed elements are going to be your red blood cells, white blood cells, platelets. Plasma is going to contain your water, plasma proteins, and their solutes. You're going to have hematocrit, proportion of blood, 45%, important number. Blood viscosity, important, resistance of blood. If the blood's too viscous, you can have turbulent blood flow and clotting, so you want smooth and fast white blood cells. Which form element contributes the most to maximum blood viscosity? Red blood cells. However, if he does say what contributes to the maximum viscosity, not which form element, it's going to be albumin. Compare plasma versus serum. So these are going to be the thing that does not sink to the bottom when you do the centrifuge. That's going to be both plasma and serum. Only difference is plasma has the clotting factors, serum does not have the clotting factors. Mean corpuscular volume is just the average red blood cell volume. Mean corpuscular hemoglobin, average mass of hemoglobin. Mean corpuscular hemoglobin concentration, average concentration of hemoglobin. So what are the functions of albumin? Plasma oncotic pressure. This is the basically the oncotic pressure that is sucking the water back into the capillary. Albumin transports lipids, bilirubin, fatty acids, steroid hormones, acid-base balance, antioxidant, and anticoagulation function. What are the functions of globulins? Alpha and beta globulins are clotting factors, and white globulins are immunoglobulins. What are the functions of fibrinogen? This is factor one of the coagulation cascade. Impacts maximum viscosity of blood, and it affects ESR proportionally. ESR is just the erythrocyte sedimentation rate. All this means is how quickly the red blood cells will fall down. And so if you have a lot of fibrinogen, you're going to have a very high ESR, because think about it, as you're putting the red blood cells in a thick solution, it's going to take more time for them to fall down. Fibrinogen is kind of acting as helping platelets attach to one another in the platelet plug. Plasma and proteins are mostly secreted by the liver, and hypoalbuminemia leads to peripheral edema. You can have decreased plasma oncotic pressure, decreased fluid absorption, fluid accumulation, tissue edema. What structural features of red blood cells? Increased surface area for diffusion, flexible membrane, spectrin anchored to band 3 by anacrin. The membrane connects to the cytoskeleton, contains hemoglobin, GLUT1 transporter, no mitochondria, and negatively charged membrane to salicylic acid. What are the functions of red blood cells? Transport respiratory gases, acid-base balance. The average red blood cell is alive for 120 days. What are three factors important for normal lifespan? Membrane must be deformable. Normal hemoglobin structure and function, osmotic balance, permeability. What happens to red blood cells after this amount of time? You're just going to have extravascular hemolysis in the spleen, bone marrow, or liver via the reticuloendothelial system. The breakdown of hemoglobin, two parts, heme and globin. The globin is going to turn to amino acids. The heme is going to turn to biliverdin and iron. The biliverdin is going to be bilirubin, liver excreted as urobilin in the urine, and stercobilin in the feces. Just remember bilin and then bilin. So urobilin, urine, stercobilin, feces. Iron via transferrin into ferritin in the liver, and then via transferrin again into the bone marrow. What's the role of G6PD and red blood cells? Generates NADPH through the HMP pathways. Anicocytosis is just variation in red blood cell size. Polycytosis is variation in red blood cell shape. What happens to red blood cells in a hypotonic versus hypertonic environment? Hypo means less solute outside, therefore it's going to swell. Hypertonic, more solute outside, therefore it's going to shrink. What hormones act to increase hematocrit? Cortisol, testosterone, EPO, erythropoietin, thyroxine. So cortisol, testosterone, thyroxine, erythropoietin. Which proteins function for iron storage proteins versus iron transport? Very high yield question. Ferritin soluble, hemosiderin insoluble, transferrin is for transport. What are three major forms of iron in the body? Hemoglobin, myoglobin, ferritin. Ferritin, hemoglobin, and then myoglobin. Three important metabolic pathways occurring in red blood cells: Embry-Meyerhoff pathway generates 90% ATP, produces DPG. Function of the HMP shunt generates 5 to 10% ATP, produces NADPH. What's the function of the methemoglobin reductase pathway? Maintains hemoglobin iron in the reduced Fe2 state and prevents the accumulation of methemoglobin, thus maintains oxygen carrying capacity of red blood cells. Erythropoietin helps in every stage of red blood cell maturation and release. Therefore, it is one of the most important factors in terms of releasing red blood cells and increasing hematocrit. Which white blood cells are granulocytes? You're going to have three of them. You're going to have neutrophils, eosinophils, basophils. The neutrophils are going to be polymorphonuclear. Immature neutrophils are going to be band neutrophils. Mature neutrophils are going to be segmented neutrophils. And eosinophils are going to be bilobed nucleus. So are basophils. Eosinophils are going to stain orange, and basophils are going to stain blue because B is with the B. Basophils are also going to be for allergic response and anticoagulation. Eosinophils are also going to be for allergic response and coagulation as well, anticoagulation. But one does have more impact, I believe, is the basophil, which are white blood cells are agranulocytes. You're going to have two of them, monocytes, and you're going to have your lymphocytes. So monocytes, macrophage, M and M. Monocytes are going to be kidney-shaped nucleus differentiated to macrophages upon leaving circulation, and macrophages do phagocytosis. Lymphocytes, just your lymph cells. So you have two cells, T cells and B cells. T cells are cell-mediated immunity, B cells are humoral immunity. Circular nucleus. Cytoplasm forms a ring around the nucleus. Need to memorize these pictures. So for your neutrophils, you're just going to have these polymorphonuclear, so a lot of nucleuses in the cell. For your eosinophils, you're going to have this weird triangular shape. This is that bilobed nucleus, kind of split in two. Then you're going to have your basophils, which are also bilobed nucleuses, but kind of think them as just one big nucleus. And then you're going to have your lymphocytes, which is going to kind of have that kidney-shaped nucleus. So to summarize, you're going to have multiple nucleuses, neutrophil. Triangle, you're going to have eosinophil. The basophil, B, big B in the B, it kind of looks like the shape of the B here. Forget about the stem. So a lot. Then you're going to have your monocytes, which is going to have the kidney shaped. And then you're going to have your lymphocytes, which is also, it's kind of like the basophil, but you still have that cytoplasm ring around the nucleus. What's the process of phagocytosis by white blood cells? Amyloid movement, diapedesis, chemotaxis, phagocytosis. It's going to move around, it's going to squeeze through the endothelial cell. Chemotaxis, attracted to the tissue. Phagocytosis, destroy the tissue. And again, this is only by neutrophils and monocytes. What's the significance of shift to the left granulopoiesis? This just means increase band. Again, band neutrophils are going to be your immature neutrophils in the peripheral blood. What's circulating versus marginating neutrophils? Circulating are included in blood cells. Marginating are not included in the blood cell count. What are the functions of platelets? Hemostasis, blood coagulation, clot retraction, platelet growth factors. They mainly just help with hemostasis, in vasoconstriction. They help with blood coagulation. They help in clot retraction. This answer can be answered in week 2 as well. What precursor site do thrombocytes come from? They come from megakaryocyte. Thrombocyte is just another word for platelet. And the main thrombopoietic growth factor is thrombopoietin. So thrombocytes, thrombopoietic growth factor, thrombopoietin. Thrombocytosis increased count. Thrombocytopenia is decreased count. Penia just means less. Cytosis just means more. What processes hematopoiesis include? 3 processes: erythropoiesis, leukopoiesis, thrombopoiesis. And what factors affect the rate of hematopoiesis? Stromal matrix, hematopoietic growth factors. So erythropoietin is the most important one. 3 stages of hematopoiesis. So you're going to have the mesoblastic, hepatic, and myeloid stage. Mesoblastic is yolk sac, week 3 to week 10. Hepatic has to be liver and spleen. So week 6 to 32, week 10 to 25. Connects into week 3 as the liver and spleen can be enlarged. in HDK. Myeloid stage, what happens? Red bone marrow from week 30 to 35. What site of hematopoiesis in adults? Going to be sternum, vertebrae, clavicle, skull, proximal ends of long bones, any important areas, pelvis, things like that. Which cell is produced first? You're going to have your red blood cells, granulocytes, lymphocytes, monocytes. Not memorizing this. What is the myeloid to erythroid ratio? It's 3 to 1, and the reason is because the red blood cells are going to survive for 120 days versus the white blood cells are only going to be for a couple of days, sometimes even a couple hours. So you're going to need way more of the white blood cells. What are the colony-forming units? They're just progenitor cells formed from stem cells. Committed cells form colonies of the cells. What changes occur as cells mature? Decreased cell size, increased cytoplasmic to nuclear ratio, chromatin becomes thick, decreased RNA. In terms of erythropoiesis or red blood cell function, when does the hemoglobin first appear? Polychromatic erythroblast, when does the nucleus disappear? Reticulocyte, when does the nucleus stop dividing? Reticulocyte as well. So here we have proerythroblast, basophilic erythroblast, polychromatic erythroblast, orthochromatic erythroblast, reticulocyte, red blood cell. So hemoglobin is first appearing in the polychromatic erythroblast, and in the reticulocyte there's no nucleus, no division. Remember, red blood cells don't have a nucleus. They're very special cells. What factors increase the rate of erythropoiesis? EPO. EPO comes up a lot. Where is erythropoietin produced? Interstitial fibroblasts in the kidneys. Very important. What effect does EPO have on erythropoiesis? Promotes every stage of red blood cells maturation, promotes hemoglobin synthesis, stem cell differentiation, release of red blood cells from bone marrow. Intrinsic factor. This is produced in the parietal cells of the stomach. It is essential for the absorption of vitamin B12. In certain conditions such as pernicious anemia, you're going to have decreased intrinsic factors, so you're not going to have vitamin B12 absorption. And so that's why in megaloblastic anemia, now that you have a decrease, or you basically have a vitamin B12 and folic acid deficiency, your blood cells, red blood cells, are going to fail to divide. And as such, they continue to increase in size, resulting in a megaloblast. And this also basically would decrease your hematocrit numbers. Now going back here, intrinsic factor. This is what is helping for the absorption of vitamin B12. Other factors that can affect the rate of erythropoiesis are folic acid, iron, vitamin B12. This is why women are actually given vitamin B12 and folic acid before pregnancy or during pregnancy to ensure there's enough red blood cells. So, or to make sure that they're not anemic, because women tend to have periods, they can have more bleeding during pregnancy, so they can actually make them more prone to anemia. Therefore, treating them with folic acid and vitamin B12 seems to be a good idea. Now, going back to the examinable content, the pathogenesis of the most common microcytic anemia is iron deficiency, and this is because you're going to have less hemoglobin during division of erythroblasts, which is going to cause smaller and paler red blood cells. Remember, hemoglobin has a very important role, and there's millions of hemoglobin proteins in the red blood cell. Normocytic anemia, where mean corpuscular volume is within range but hematocrit is decreased. So, by definition, megaloblastic anemia cannot be a normocytic anemia, because a normocytic anemia means that the volume is the same, but the hematocrit is decreased. Megaloblastic is where your MCV would actually increase, and you also have anemia, which means there's a decrease in hematocrit. Anemia usually just references to decrease in hematocrit. However, the definition itself has to do with proportion of red blood cells, and sometimes can also do with iron, but the best way to think about it is anemia, decrease in hematocrit. So normal chromic anemia. What is normal? The hemoglobin concentration is normal. However, you're going to have less red blood cells. Why is this case? Maybe you have a problem with the production of red blood cells, maybe you have too much destruction of red blood cells, or Maybe you just don't have enough release. Many different mechanisms that play there helps connect them in week 4, I believe. Hematostasis and clotting pathways. Hematostasis, what does that mean? It's just the stoppage of bleeding. And you're going to have two phases, your primary and your secondary phase. The primary phase is going to be very fast. It's going to be short-lived. It's going to involve vasoconstriction. Um, and then it's going to be essentially, you're going to have a platelet plug in there as well. For secondary hematostasis, you're going to have coagulation factors. You're going to have a fibrin clot form. It's going to take more time. Hence, you have the primary hemostasis there. And secondary hemostasis is going to be long, and it's going to be initiated by exposure of tissue factor on cell membranes. Now, the thing with primary hemostasis is you're only going to have vasoconstriction if that if that vessel has smooth muscle. You can't have vasoconstriction if the vessel doesn't have smooth muscle. Now, going into the 3 stages of hemostasis, you're going to have vasoconstriction, platelet plug formation, and you're going to have clot retraction. That's true, but again, you also want to talk about platelet plug formation. That's a component of hemostasis, so there technically should be 4: vasoconstriction, platelet plug formation, clot formation or coagulation, and then clot retraction. That is the answer, not primary, secondary, if that's asked in a question. So, what are some factors that can act as vasoconstrictive agents? Thromboxane A2, serotonin, epinephrine, endothelin. Very easy to remember. Thromboxane A2 can be said for almost anything. Epinephrine, you get scared or you need to run quickly. Think about it like that. You damage yourself while fighting someone. You're going to have norepinephrine and epinephrine in you. Serotonin is also interesting. Usually something that's used to relax, but nevertheless, it stands there. Platelet plug formation. Platelets are going to be activated by cytokines and contact with the collagen. Become sticky and attract more platelets. Platelet plug form. Very oversimplified. You're going to have subendothelial collagen, which is going to attract the 1-Willebrand factor in the circulating blood. The 1-Willebrand factor is then going to touch GPIA, or GPIB, sorry, and that is going to then attract the platelets, and then they're going to have this cycle which keeps on attracting more and more platelets until a plug is formed. And then you're going to have fibrin. Fibrin is like the glue that can help make the platelet plug very strong, and is essentially what stops the bleeding. Then you're going to have clot retraction, because what this mechanism is essentially a positive mechanism. It's a positive feedback loop. It will keep on going and going and going. And that's not a good thing, because if the clot is too big, the clot can actually split off. It can go to your brain. This can form a stroke. Or, contrary to that, the clot can just be too big and it can occlude the blood vessel. So clot retraction seems to be a very important part. Naga mentioned in Piazza that prostacyclin actually does not have anything to do with clot retraction. So, GP2B and IA links cytoplasmic actin filaments to fibrin. Activated platelets signal the formation of actin binding proteins. Still, not really clot retraction. The most important thing of clot retraction is platelet and myosin contraction brings about the retraction of the clot. This is what you need to know. What does that mean? That just means that the myosin, or the muscle, if you want to think about it that way, is just contracting and it's just squeezing the fluid out of the clot. And this is what helps in clot retraction. Notice prostacyclin decreases aggregation, or the initial platelets clumping together, but prostacyclin does not involve in clot retraction. Now, for platelet factors, basically these are just going to be alpha granules. Remember, that is a type of globulin right there from week 1. Alpha and beta granules are globulins. Why? Globulins are immune factors. Now, for the alpha globulins, 1-Willebrand factor, factor 1 fibrinogen, factor 5, and factor 8. Factor 8 is antihemophilic A factor. Factor 9 is Christmas factor. I forgot what factor 5 is, I believe. Factor 5 is not needed to be known, but you need to know fibrinogen factor 1, prothrombin factor 2, tissue factor factor 3. Factor 4 is calcium, and that is all you need to know. Now I will put in the MCQ just the factor name, or you'll put antihemophilic A, and you need to know that that's factor 8 in the MCQs. So this would be factor 8 right here. What factors release from beta granules? ADP, serotonin. Now, what factors are released from other granules? Phospholipids, actomycin, thromboxane, thromboxane A2, thrombodenin. What are the 3 steps of platelet bulk formation? So you're going to have adherence, or kind of circulating and getting all the things that you need. You're going to have activation, so activating the things you have collected, and aggregation, which they're all just clumping together. That should help quite a bit. So adherence, von Willebrand factor circulating in blood, it's going to connect to the subendothelial collagen. It's going to bind to the GP2B, von Willebrand factor. It's going to bind to GP2B, and it's going to link the platelet to exposed collagen fibers. And then your GP1A is just going to directly link the platelet and collagen. Again, GP2B is going to bind to von Willebrand factor. GP1B is going to bind to von Willebrand factor. The GP1B is then going to touch GP2B, which can then bind to the platelet or expose collagen fibers. And then your GP1A can link to the platelet and collagen. It kind of leads to a cycle. Activation, what happens? Platelets are stimulated by ADP and thromboxane A2. Aggregation, what happens? Fibrinogen cross-linking joins platelets through GP2B, GP2A forms plug. GP1B, then you're going to have GP2B, GP1A. What positive feedback loop helps platelet aggregation? So ADP is what is essentially activating the platelets, and then the activated platelets release more ADP. So you're just going to have this positive cycle. What effect does cyclic AMP have on platelet aggregation? Very simple to understand. If you increase intracellular or cytosolic calcium, you're going to increase the release of certain chemicals. So thromboxane A2 increases cytosolic calcium. We know that it increases aggregation. Cyclic AMP is just the opposite of that. So thromboxane A2 increases cytosolic calcium. It's going to decrease cyclic AMP. Prostacyclin decreases cytosolic calcium. It's going to increase cyclic AMP. It's the best way to learn that. What factors inhibit platelet activation? Prostacyclin, NO, nitric oxide, something like that. Flowing blood. Again, if the blood is flowing, you're not going to have the clots. If it's turbulent, you're probably going to get clots. What are the 3 groups of clotting factors? Surprisingly important. Factor 11, 12, prekallikrein, HMWK. These are your contact factors. They're stable in blood. They do not need calcium. Then you're going to have your vitamin K dependent factor groups. Warfarin is going to inhibit these factors. You're going to have factor 2, factor 7, factor 9, and factor 10. Factor 8 is here, fibrinogen group. So fibrinogen group is going to be factor 5, factor 8, factor 13. It's going to react with thrombin. It's going to be completely consumed. And it's going to increase during pregnancy. That is why pregnancy is a hypercoagulable state, because you have increased fibrinogen factors. What are the steps of the intrinsic pathway? The intrinsic pathway is quite simple. It's going to be factor 12, 11, 9, and then your 9, 8, calcium, PL formatinase. How do you remember that? Well, in the extrinsic pathway, E is for extrinsic. 3 also has an E in it. 3, 7, 10. 3 plus 7 is 10. Therefore, you cannot have your 3, 7 is 10. Therefore, you can't have the 10 in the intrinsic pathway. And you start off at 12. So if you're going from 12, 11, you skip 10, you go to 9, and then you're going to essentially have factor 9, 8, calcium, PL form a tenase. Quite easy. 3 plus 7 is just going to be the 10. The tenase is going to activate 10. So both the intrinsic and extrinsic pathways are going to activate factor 10. So for the extrinsic pathway initiated by tissue factor 3, 3 activates factor 7, and then just 3, 7A calcium form a trimolecular complex which is going to activate factor 10. Now once you have activated factor 10, you're going to have basically these two pathways, and the two pathways converge to one common pathway. That's why it's called common. And you're going from prothrombin, which is factor 2, to thrombin, and it can be activated by a lot of things. Factor 10A is just the activated form of factor 10. It can be activated by calcium, factor 5A, in the presence of 10A, and PL. So prothrombin is to thrombin, and then the thrombin is going to convert the fibrinogen to fibrin. And the fibrin is important because, remember, factor 1A, this is important because it basically makes the soluble clot to an insoluble clot. By factor 13 here also helps with that conversion from fibrinogen to fibrin. I guess thrombin is also a big part of that as well. Now, what are the functions of thrombin? Well, we know it's going to have a positive feedback on factor 8 because it said here that factor 8 would have to be in the intrinsic pathway, I believe. And yes, factor 12, 11, and then your 9, 8 calcium is going to form the tinase. It's going to be an intrinsic pathway. So thrombin seems to have an increase on factor 8, so thrombin would impact the intrinsic pathway as well. However, interesting, coagulation depends largely on the extrinsic pathway. It's going to impact factor 5, factor 7. This is the extrinsic pathway right here. And factor 11 catalyzes the formation of cofactors 5 and 8 and activates 8 to 8A. Again, this is the intrinsic pathway here. Causes endothelial cells to release ADP, 1 bilirubin factors, NO, PGI2, and tissue plasminogen activator. Activates platelets to release platelet factors. Cleaves factor 11 to 11A on the platelet surface. Prevents excess clot formation. Activates protein C via thrombin system. The key things to know is thrombin is important for 3 main reasons. You're going from prothrombin to thrombin, so it has to be there. It has to be there in terms of helping with your fibrin to fibrinogen, I believe. Fibrinogen to fibrin, my bad. So fibrinogen longer to shorter, fibrinogen to fibrin, which is important to form the insoluble clot. Thrombin is also important because it releases platelet factors, and then contrary to all this coagulation, it also releases activated protein C via the thrombomodulin system. How do all these pathways interact with each other? The extrinsic tenase activates factor 9 and factor 11 of the intrinsic pathway. So the tenase that you form with 3, 7, and then 3, 7, 8, 10, it's going to activate factor 9. Factor 9 is important because remember, you're going 12, 9, and then you're going 9, 8A of the 8 calcium, PL of the intrinsic pathway. Then your factors 11 and 10 of the intrinsic pathway activate factor 7 of the extrinsic pathway. And then your thrombin is catalyzing formation of cofactors 5 and 8A. Again, 8A is going to be your intrinsic pathway. And coagulation is largely depending on the extrinsic pathway, interestingly. Why is coagulation increased in inflammation? Inflammation is going to trigger monocytes to express factor 3. Again, this is going to be the extrinsic pathway. This is going to increase the risk of thrombosis and sepsis. Why is pregnancy a hypercoagulable state? Well, we already know that there's going to be an increase of majority of clotting factors. Why is this? This is because you can have hormonal changes due to increased risk of thromboembolism, which basically is just like a lot of clots forming. Again, you have a reduction in fibrinolytic activity, so you don't have a lot of breakdown of the fibrin, and that's important because fibrinogen to fibrin is helping with the insoluble clot formation, and then you have a decrease in natural anticoagulants. Why do you have that? Childbirth often has a lot of bleeding, so the body is going to reduce these clotting factors in order to accommodate to this physiological change. Anticoagulation. Why is anticoagulation important? It's going to prevent clots from occluding vessels and emboli forming. What does tissue factor pathway inhibitor do? This just binds to the trimecylar complex. So again, that's going to be the extrinsic pathway, and it blocks the protease activity of factor 7. We know that the extrinsic pathway is the most important pathway when it comes to this coagulation cascade, in terms of converging to the common pathway and forming that insoluble clot. So therefore, tissue factor pathway inhibitor probably is going to inhibit the extrinsic pathway. Thrombomodulin. So this complexes with thrombin, removing thrombin from circulation. So thrombomodulin is removing thrombin. Again, we know that thrombin is going to promote those clotting factors. Therefore, thrombomodulin should inhibit coagulation, and it's going to be activating protein C. Thrombin would also be activating protein C, but I guess thrombomodulin probably just activates more. What does protein C and protein S, they bind to thrombomodulin in its little complex, and basically just inactivates factor 5 and factor 8. Again, factor 8 is going to be your intrinsic pathway, and that just inhibits coagulation. Intrinsic pathway, playing table tennis, indoors, intrinsic. So that would actually be increasing your PTT time versus playing tennis. You play tennis outside, extrinsic. Therefore, your PT time has to do with the extrinsic pathway. Now, what does antithrombin 3 do? It inactivates thrombin in factors 9, 10A, 11, 11A. So probably more to do with the common pathway, I guess both the intrinsic and extrinsic by extension. Factor 9, that's going to impact your intrinsic pathway, and factor 12, that is going to be also the intrinsic pathway, activated by binding of heparin. So heparin leads to antithrombin, inhibits just basically a whole lot of intrinsic and common pathway, and it would be, I guess, decreasing your clot, or decreasing the chance that you're going to have that insoluble clot form. What does the fibrinolytic system do? This is going to break down the fibrin mesh that is formed in the clot. And remember, fibrinogen to fibrin. Fibrin is the thing that is making the clot insoluble. Fibrin is degraded by plasma, which forms from plasminogen by calcarein and factor 12. Remember, factor 8 and factor 9. Factor 9, Christmas factor. Factor 8 is antihemophilic A factor, are different. This is calcarein and factor 12. Now you have your extrinsic activators. Tissue plasminogen activator, urokinase type activator. Wasn't covered in lecture or even the GLS. So there's that. What does warfarin do? Super important. Warfarin inhibits production of vitamin K dependent clotting factors, decreases prothrombin and other procoagulant factors. Important. Warfarin inhibits vitamin K dependent clotting factors. What does aspirin do? Inhibits platelets enzyme cyclooxygenase and inhibits thromboxane A2 synthesis. Thromboxane A2 is important for aggregation, so it's going to inhibit platelet aggregation and increase bleeding time. What does bleeding time test? So bleeding time is just your primary hemostatic response, right? What conditions are going to increase your bleeding time? Thrombocytopenia, which means you don't have a lot of platelets. Anything to do with von Willebrand deficiency or the platelet count. Von Willebrand disease, good example. That will increase your bleeding time. What is coagulation time? This is essentially testing your secondary hemostatic response. And it's the time from initiation of bleed to formation of that fibrin insoluble clot. And what conditions would basically increase the clotting time? Hemophilia A and hemophilia B, factor 8, factor 9. Clotting factor deficiency, vitamin K deficiency. Liver disease, you're just going to have decreased overall clotting factors, DIC and VWD. Don't really need to know these two. What does prothrombin time? Again, playing tennis, extrinsic pathway. So time for formation of a clot when 3 and calcium are added to blood. Remember, extrinsic pathway, factor 3 is going to be exclusive to the extrinsic pathway. So if we add it to blood, we can basically test the extrinsic and common pathway time. And what conditions would increase prothrombin time? So remember, vitamin K deficiency, warfarin therapy, liver disease, DIC. And this kind of hints because of vitamin K factors. If we see vitamin K clotting factors. Yeah, so you're going to have your factor 7, which is your extrinsic pathway. You're going to have factor 9, which is technically intrinsic, protein C, protein S. I guess factor 7, that'd be really easy because you know that vitamin K deficiency, warfarin, it would increase PT time. Now, what does activated partial thromboplastin time, APTT test? PTT, playing table tennis, we play that indoors. Therefore, it is the intrinsic and common pathway. And what conditions would increase APTT? Hemophilia A and B. Why? Because you have a deficiency in factor 8 and factor 9. Factor 9 and factor 8 are part of the intrinsic pathway. Factor 12, 11, and 9. And then you have your 9, 8, calcium, PL. Therefore, APTT or PTT, as it's also known, would be increased if you had hemophilia A and B. Vitamin K deficiency, kind of here as well. Remember, we had that common vitamin K factor. Heparin disease, liver disease for both because it's just a decrease in overall factors, and then DIC. Week 3, really quickly. Before we do that though,